Okay, welcome back friends. Welcome back in the another video lecture of our viral transduction series. So today we will going to discuss about adeno associated virus. So adeno associated virus, as its name suggests, it required the association of adeno. That means adenovirus. So let's see what it is. So adeno associated virus, AAV, I am referring it like this. So this AAV have the ability to transduce a broad range of cell types. Okay, but there is only one problem, that it require the association of adeno or another kind of virus, let's say herpes virus, for the production of the recombinant virion in the packaging cell line. Okay, and that is why you cannot obtain a high quality viral stock which will not contain any kind of helper virus. That means high quality viral stock free of helper virus is cannot be obtained in case of adeno associated virus. Okay, so adeno associated virus have the ability to transduce broad range of cell types, no doubt about this, but it required the help of helper virus, that can be either adeno or herpes virus, to produce recombinant virion in the packaging cell line. Okay, and that is why you cannot get a high quality adeno associated viral stock which is devoid of or lack of helper virus. Okay, so furthermore, adeno associated virus have a very relatively low packaging capacity, that is 4.9 kb. This is most probably the lowest up to that part that we have discussed, because in lentivirus we have seen 9 kb insert size, and in retrovirus we have also seen kind of 9 kb, that is 8 kb, and in adenovirus we have seen 7 to 8 kb. That means this adeno associated virus has the lowest insertion capacity, that is 4.9. maximum titer 1 into 10 to the power 11 particles per milliliter, and I am again saying this is not pure adeno associated virus. Okay, there can be presence of also helper virus. Okay, and it have the ability to infect both dividing and non-dividing cell. So expression, stable site specific integration, that is the important thing that I want to mention you, that adeno associated virus have the ability to transfect human cell in such a way that it will integrate in the human chromosome at specific site. One more time, adeno associated virus, after entering into the human cell, it will integrate its genomic material at specific site, okay, in the human chromosome, and this is known as site specific integration. So, and this phenomena bypasses the chance of insertional mutagenesis that we are facing in the other type of viral vector we have discussed until now. Okay, so this is the major advantage that we can get in case of adeno associated viral delivery system, but it has a major limitation, that you have to co-transfect your adeno associated virus with another helper virus, for example your adenovirus or herpes virus, that will help them to produce a recombinant virions inside the packaging cell line. Okay, that is why viral stock of pure adeno associated virus is not possible to derive. So again, let's revise that adeno associated virus have the ability to transfect both dividing and non-dividing cell. That means it also have a wide coverage. Now this adeno associated virus has only one problem, that it required helper virus such as adenovirus, that is why its name adeno associated, and herpes virus for the production of the recombinant virion into the packaging cell line. Okay, so now the insertion capacity is relatively very low, that is only 4.9 kb. On the other hand, adeno associated virus have the ability to elicit a only very weak viral immune response. That means it show a very low immunogenicity in many cell types. That means we have not bothered about, we do not have to bothered about the toxicity, right? Because here the cell will not consider the adeno associated virus as foreign invader. Okay, so that is a good news. Along with another good news is it prevents or it bypasses the insertional mutagenesis possibility. So total size is also relatively low, right? Because in other cases we are getting near about 9 or 11. For example, this one we have getting 10, and in case of retrovirus we most probably seen up to 11, right? So, and the very first one we have seen near about 36. That is quite high. So adeno associated virus have the lowest insertion size and lowest total size also, right? The maximum titer 1 into 10 to the power 11, although it is not pure adeno associated virus. Infection, dividing, non-dividing cell, expression, stable site-specific integration, and required co-transfection with a helper virion for the production of the recombinant virion in packaging cell. Difficult to obtain high quality viral stock that are free of helper virus. Okay, so that is the ending of that lecture. So before I will wrap up this lecture, let's one more time revise it. So adeno associated virus have the ability to transduce a wide range of cell type that can be both dividing and non-dividing. It has only one problem, that adeno associated virus require helper virus to produce or in the production of the various kind of recombinant virions inside the cell, right, inside the packaging cell. Okay, that is why you cannot get a high quality viral stock that lacks or that do not contain any kind of helper virus. Okay, that means pure viral stock isolation of adeno associated virus is not possible. So insertion size, that is our DNA insertion size here 4.9 kb. So total size of the DNA is 8.5 kb, and particles, that is the maximum titer is 1 into 10 to the power 11 particles per milliliter. Okay, and the most important advantage is it has a relatively low immune response. Okay, that means it will generate only a very low immune response in various cells. That means those cells will not consider adeno associated virus as a foreign invader, right? That is quite good news. Along with adeno associated virus integrates into the human genome at specific site. That means it bypasses the possibility of insertional mutagenesis. Okay, so that was the concept for adeno associated virus. So I hope this video will be helpful to you. Thank you.